Good evening. First, we wanted to say thank you to everyone for coming. We know in the thick of having just finished Thanksgiving holiday and prepping for final exams, it's been a bit hectic, but we couldn't pass up this opportunity to recognize an incredible professor.
For those of you who don't know what E4 is, it's an annual event that we have here where we recognize the professor and he gives a speech on a given topic. So this year it's the milestones that make him a better engineer. And yeah, we're really excited. We kind of chose this topic because yes, engineering is about academics and solving complex problems and all that wonderful stuff. But it's also so much more than that. It's oftentimes those unexpected milestones and lessons that really shape us to be both who we are as people and engineers. So without further ado, please welcome our 2018 E4 lecturer and awardee, Dr. Phelps.
Yep. All right, cool. And this comes off, right? Maybe. There we go. All right. Wow, so I have to say I'm really honored and I was a little shocked actually that I was asked to do this because I'm actually a pretty young professor. I just started here at UF 2 years ago. I'm an assistant professor. So when I got the email that said the last lecture, I was like, wow, geez, I hope it was more like the first lecture, right? I'm gonna get hit by like a bus on the way out of here or something. So yeah, I shouldn't say that, but really I was really amazed because, you know, I figured you would get some like wise, grizzled old guy that's been around and has a lot of life experience to tell you about all of his wisdom or her wisdom. And so for me, it's a huge honor that the undergraduates asked me to do this. So anyway, thank you all very much for having me. Okay, so the topic you charged me with speaking about today is the unexpected milestones that made me a better engineer. And I've spoken at this at a couple of events around campus. I usually do a guest lecture for the intro to biomedical engineering class, and I might suspect that that's where somebody heard me talk about some of these things. So hopefully I've got a few stories to share with you guys that you'll find interesting and maybe a little bit funny too. Okay, so I'm going to give you, here's an overview of the topics I'm going to address tonight. Where am I now? How did I get here, and where am I going? Those are pretty basic questions, fundamental questions that everyone has pretty much about what they're doing at any stage. So let's start with the first point. Where am I now? So who am I? I'm an assistant professor of biomedical engineering at the University of Florida. That's me. This is my lab, one of my Ph.D. students working at the microscope together. For some reason, University of Florida thinks that I'm fit to trust with a budget, expensive medical equipment. It gets delivered in crates. A few people have given me awards for my research, and some of my things that I've done have been published in pretty nice journals and featured on the cover. So how did I get to this stage? Well, first I want to address the topic of biomedical engineering. Are there any BMEs out there in the room tonight? Okay, a handful. Okay, anybody who's not a BME? Yeah, great, most of you, good. Okay, so BME is a super new discipline. It's the new kid on the block in terms of College of Engineering. And biomedical engineering, what it is, it's the application of principles and problem-solving techniques that we all learn as engineers to problems in biology and medicine, right? So BMEs or biomedical engineers, we differ from the other engineering disciplines in that we use and apply biological principles in our engineering design process. So that's combined with aspects from mechanical, electrical, chemical engineering, material science, biochemistry, and so on, computer sciences as well. So it's really a jack of all trades as well. It's very broad. It covers a lot of topics. So I kind of want to give you some insight into what it is that I do, what aspect of biomedical engineering is important to me. All right. So what I study in my lab is the pancreas. So I brought a pancreas with me. Here's one. It's a plush pancreas. Here, you can pass it around. Okay. All right. That's about the size of a real human pancreas. And so human pancreas is an organ. Everyone quick point to your pancreas. Where is it? Right? Somewhere in here. Some obscure organ. It's like the spleen. No one knows where the pancreas is. Okay, the pancreas sits right behind the stomach. And what does the pancreas do? Does anyone know? What about sugar? regulates your blood sugar. Yeah, that's what it does. So it does that through a hormone called insulin. So the pancreas, one of its jobs is to secrete hormones, insulin and glucagon, that regulate blood glucose levels, right? The other thing, it does something else too. What else does it do? It's really two organs in one. The other thing, anyone want to guess what else you need a pancreas for? All right. So it makes something that we call pancreatic juice. That's kind of gross sounding. And what pancreatic juice is it's a mixture of bicarbonate, and so you need bicarbonate to neutralize acid that's produced in your stomach, and it also has a lot of enzymes to help break down the food that you eat and your digestion. So it does two different jobs in one. So this is a cross section of a human pancreas. If we cut it down the middle. And look inside, you can see that there are these red spots, okay? And those are the little patches that we call endocrine tissue. In fact, they're actually called islets because they look like islands. And islets of Langerhans, for the scientists to discover them, Paul Langerhans. So that's what I'm interested in, these little red areas. And that's what we call the endocrine tissue. That's where the hormones are made. So all the other stuff outside is where all the digestive stuff is happening. So if we zoom in on those pancreatic islets, they're made up of different cell types. You have, they're colored here. The red ones in this picture are beta cells. Those are the cells that make insulin. The green cells are alpha cells. Those are the cells that make glucagon. And insulin and glucagon are antagonistic hormones, meaning they work in opposite directions. Insulin lowers blood sugar when it gets out of range, and glucagon increases blood sugar when it gets too low. So that's what we call homeostasis, keeping it within a range. Now, somatostatin is another hormone that's being produced that helps control all of this process. So these are three different cell types that are in there that all have a different job. Now, you notice that the human islet is really different from the mouse islet, right? Yeah, the architecture is different. So humans are not mice. We use mice in research a lot to try to model processes. But I do a lot of research actually using human tissues in my lab. Okay, so one of the things that we're interested in is engineering tools to better understand beta cell function. And so this is a diagram of how beta cells work. How do they secrete insulin in response to an increase in glucose? They have a special transporter called GLUT2. And what happens is glucose gets transported inside the beta cell. Some of you guys have had biochemistry, right? Hopefully? Yes? A few of you. And so you know about ATP, right? So you make ATP. This is the energy molecule inside the cell from glucose. And ATP allows you to store energy, and it gets burned to do different processes in the cell. So the glucose is coming in, you have respiration, you're making ATP, and then as the ATP level rises in the cell, it closes these special channels called potassium channels. And you get a depolarization of potassium ions. So anyone ever heard of an action potential before in a neuron? Yes? Right? So this kind of works like that, where you have an electrical depolarization. This is the cell membrane. It's actually an insulating material. So it separates charges inside and outside the cell. So they build up on this side, you get a depolarization wave, and then there's another channel called a calcium channel, voltage-gated calcium channel, and that opens up, and calcium pours into the cell, and that's the green, and then the insulin is stored in these little granules, and that merges with the plasma membrane and dumps outside. All right. So that's a complex process, right? So one of the things we do is we try to observe these kind of things happening live in the lab. So this is a preparation of beta cells that we've infected with a virus to make them express a fluorescent protein that's sensitive to calcium. So the brightness of the protein gets brighter or darker depending on how much calcium is present. So we can actually look at this process, this green calcium coming into the cell. So what happens is when the beta cells are in low glucose, there's no calcium as you would expect, but when we add high glucose, they start responding. So I'll play the video here. In low glucose, they just sit quietly, and then in a second, here comes the high glucose, and they start responding. Look at that. Wow. To me, this is super cool stuff. I love this stuff. I geek out on it, right? I think it's really amazing to see these guys. They're responding. What else can you notice? You can see they're electrically connected to one another, right? The signal jumps from cell to cell. At that point, we add a drug called diazoxide that shuts down the whole process. And then at the end, we add potassium chloride, which makes them all light up together at the same time. So they're connected. They pulse in unison. You have these kind of lightning bolts that go around. All right? So that's how the beta cell works. So this is a picture of 2 human beta cells that I took in the lab on one of our microscopes. And this gives you an idea of what is going on with the insulin secretion. So these are microtubules. That's a cytoskeleton. So those of you guys that are in civil engineering, structural people, this is what gives the cell its mechanical strength. Okay? It gives it shape. It gives it tensile and elastic strength, all the different components of the cytoskeleton. So the insulin is in these little dots. I also love this picture because it's like UF colors, right? Orange and blue. So these are insulin granules, and they're packaged like that, and they get released and dumped outside of the cell. So anyway, just to give you an impression of how many little things you have in these tiny cells. This is only about 10 microns in diameter, so very small. All right, so the thing that's really near and dear to my heart, what we really study in the lab is a disease of the beta cell of the pancreas called type 1 diabetes. Type 1 diabetes is an autoimmune disease when your own immune system attacks those beta cells and those islets and eliminates all the cells in the body that make insulin. All right, so what you're seeing here is a human pancreas in the process of an islet in the process of being destroyed by the beta cells. So the T cells here are in green. T cells are the immune cells. The beta cells here are in magenta, and those T cells seek out and destroy those beta cells. They see them as foreign. So that's not something you want to have going on, and it's not normal. And what it results in is someone with type 1 diabetes can't make any more insulin. So that's what we study in the lab. Okay, and now on top of that, we try to come up with and engineer strategies to reverse that process. So it's what we call immune tolerance. So it's like the reverse of a vaccine. So this is what I was just showing you. You have all those T cells coming in, attacking an islet, killing off the beta cells. So we want to halt that process and stop it. And we want to do it in what we call an antigen-specific manner. And what that means is that we only want to stop the T cells that are attacking the islet. We don't want to stop all the T cells. Otherwise, we would immune suppress the patient. Okay? So this is the kind of problems we work on. In addition to that, if you take someone that has type 1 diabetes, they've already lost all their beta cells, so we kind of try to come up with ways to replace them. So that's a procedure called islet transplantation. We can actually take donor islets and put them into a patient with type 1 diabetes, and we try to design materials that augment that process. So this is a platform that I developed that's based on polyethylene glycol. It's a polymer. This is a chemical reaction where we cross-link 4-arm macromers of polyethylene glycol that are N-terminated in maleimide groups. That's a sulfur-hydro reaction for you chemical engineers out there. Okay, this is what we call a Michael type addition. And these 4-arm chains get cross-linked into a network, and then this creates a really nice, soft, squishy material that we link in bioactivity as well to interface with the beta cells. Okay, so I also teach. This is the class that I teach, BME 3060, Fundamentals of Biomedical Engineering. And so this is a sophomore level class. And this class I really love this class. It's taught me a lot learning how to teach. No one is really born knowing how to teach, I think, when you start out. It's very hard. And you slowly get better at it. The students teach you a lot. Like the feedback you get, their reactions to you is very important. But the concept that's very important to me personally, I have 2 little daughters, 2 years old and 4 years old, so I watch a lot of Disney movies at home. It's from the movie Lilo and Stitch, which is the Hawaiian word ohana means family, and family means no one gets left behind or forgotten. So that's my approach I take in my teaching, is I try to reach all the students in my class. Okay, so then I have my lab. My lab is my second family, all the students in my lab. We get out and do stuff in the beautiful nature all around UF besides working at the bench. Okay, so how did I get here? Straight arrow, right? Starting 2002 as an undergrad. 4 years of undergraduate, 6 years of graduate school, 5 years postdoc, 2 years as assistant professor. Hopefully if I don't bomb too badly tonight, they'll hire me again next year. I'll maintain my appointment. Okay, so you can measure this in different ways. These dots, the open circles are undergraduates that I've advised. The closed circles are graduate students that I've advised. You can see I'm getting more and more as I become an independent PI. These are papers that I've published. Okay. Color corresponds to the stage of my training. So you can see even after I finished as a Ph.D. student, still things are coming out 2, 3, 4 years later. It takes a long time to publish your work that you do in the lab. Maybe you could measure it by citations. How much influence do I have in my field? So that's another metric. Or you could measure it by teaching. So the stars are classes I've been teaching. So I have a lot heavier teaching load now that I'm a professor. Or what about grants, grant funding? Okay. Dollar signs, those are little awards, big awards that I've gotten over the years. So what do you notice about this timeline now, right? Back here, I had a lot of free time, right? I could take advantage of pursuing things that were interesting to me. I could take classes, I could do study abroad, I could think, right? I could watch Netflix. I don't think we had Netflix back then. And as time went on, Netflix back then, you came in a little envelope in the mail. You ordered one DVD at a time, right? And now, right, there's no time at all. So I have to pick and choose what I do. But my advice to you guys is that are in this stage of your life, take advantage, right, of the time that you have. Make sure you go out there and that thing that you're wanting to do, go do it, right? Because this is the time when you have time to do it. It's really important. So this is how I pretty much feel every single day. How old do you have to be before you know what's going on? Okay. How many people have seen this before? Iceberg illusion. Yes, most of you, right? So I don't need to like play it all out for you, but the general point is, normally I stop my talk right here. I give a lot more detail on the research, and people see this part of me, right? But what I'm going to talk to you guys about tonight is this part of me, right? The stuff below water. So you guys get to see the stuff that most people don't. So where did I come from? I came from a little place called Oak Ridge, Tennessee. Beautiful place in between two different mountain ranges. Over here you have the Appalachians, and then this is the Cumberland Mountains. A lot of water, a lot of ridges, Tennessee hills. But not directly. So I was actually born there, and then when I was 2 years old, I moved to California with my family, and then we moved to Washington, D.C. My brother was born, then we moved overseas for a while, and then we moved back to California, okay? Then we moved back to Tennessee again. So actually what I tell people is when they ask me, like you sit down in the restaurant, the waitress is like, where'd you come from? I say, I'm from Tennessee and various other places as well, all right? And throughout the rest of my life, I've moved around quite a lot. So that's thanks to my dad, this guy right here. Any civil engineers out there? Yeah, a few? Good. So my dad's a civil engineer, actually. He gets to wear a hard hat to work. I wear a lab coat to work. But he's a really special guy to me. He's worked for a company called Bechtel for his whole career, which is a big civil engineering company on big construction projects. He just retired and we just got the Lifetime Achievement Award from the American Society for Civil Engineers for his work. So, he was a great role model for me growing up. And that's my mom and my brother. My mom's a special education teacher. So then this is my grandfather. My grandfather is a physicist. He actually taught physics at the U.S. Naval Academy. And then that's me with hair when I was much younger. You didn't recognize me, right? Yeah. Okay, so Oak Ridge, Tennessee. Oak Ridge, Tennessee. Anyone heard of it before? Maybe? History class? Yeah. Oak Ridge, Tennessee is a really interesting place to be from because it's a Manhattan Project town, a secret city, right? So these are all the Manhattan Project sites where various secret things were going on for the generation of atomic weapons during the Second World War. These are some of the facilities at Oak Ridge, Tennessee that were really foundational in physics. This is the uranium enrichment plants X10 and Y12, and this is the second nuclear reactor ever made. This is a graphite reactor. So any of you have ever studied the history of this, you heard about the Chicago pile that they made underneath the stadium at University of Chicago. That was the first nuclear reactor. This was the second one in Oak Ridge. So this place wasn't even on maps for a long time. But nowadays, it's a little bit different. It's a normal place. There's a huge national laboratory there that does all kind of amazing research. We have a Peace Bell and a Sister Cities program with Japan. This is our sister city in Naka, Japan. And I want to give a little shout out to my high school. Oak Ridge High School was actually the first high school in the South to become integrated after Brown versus Board of Education in 1954. Most people don't know that. I didn't even know that when I was going there. And the reason is because Oak Ridge was still sort of a federally owned enclave at that time in history. And so there wasn't like a state board of education to deal with it for this particular city. Okay, so a moment that sparked me into science. 8th grade science fair, 1996. Does anyone know what that thing is? It's a little worm. It's called a planarian. It's a flatworm. They live in fresh water. They're really tiny. The interesting thing about these guys is if you cut them in half, they regenerate. So you cut it in half, the tail grows a new head, and the head grows a new tail. Yeah, it's crazy. In about 2 weeks. So this is my 8th grade science teacher, Pat DeRuse. So I did it. She was an amazing teacher. She made us do a research project in 8th grade as part of our science class. And so the project I was working on was these planarians. We cut them in half, grow them back. And I thought, hey, wouldn't it be cool? Because these things have memory, actually. They have a nervous system. You can train them to go through a T-maze with food. So they always go right or left through the T-maze. So I said, what if we train them to always go right, and then we cut them in half, and we see if with the tail regrows its head, if it remembers which way it went, right? So I did it, and it worked, right? I had like hundreds of little worms going in there in my bedroom. My parents were like, what are you doing with this? Right? Oh, this is disgusting. And I was like, no, I'm training them. Like, you're training worms, really. I'm like, yes. Okay. And then you're cutting them in half, yeah. So, but I did it, so I got all my results, and I made my little poster, right? And I go to the science fair, and the science fair judges come up to me, and they're like, really? this doesn't, why don't you go do something grounded in reality? This doesn't make any sense, right? There must be something you didn't control for. Like they're sensitive to light. The light is on the side coming from this way or that way. So they basically, I got like last place for this project. I mean, they said, okay, it's fine. Like the poster looks nice, but you know, and I was like devastated because I worked super hard on this. But I was like, okay, fine. I go on with my life, right? So in 1996, July 2003, I'm sitting at my computer and this pops up. This pops up in my newsfeed. And I'm like, what the heck, right? Yeah, so anyway, even if people tell you you're wrong, you might be right. You should believe in yourself. Okay. So this is my 12th grade AP Calculus teacher. I also have to mention her, Benita Albert. I've got some notes on her because it's super important. So she's one of those teachers of student legend. She was by far the most talented and effective teacher I've ever had. Okay? She spent 42 years in the classroom. She won the Presidential Award for Mathematics Teaching. My high school BC Calculus class, 100% of the students earned a 5 on the AP exam. Okay? Can you believe that? Right? Yeah, insane. She was that good of a teacher. So how did she do it? Just basically relentless passion and dedication. She made herself available 5 a.m. office hours, and people went to them, right, before class. Yeah, okay. So she cared about investing in the success of other people. And to me, that's really an important lesson. I just mentioned one other thing I did. I'll show you some pictures quite a lot through this presentation. I took a photography class in high school. I think engineers have an artistic side to them. I think it's really important to exercise that part of your brain. And to me, this was sort of groundbreaking for my psyche to learn about optics and how to do all the chemistry of the darkroom and how the cameras work. And now I'm super fascinated with microscopy. I think it's directly because of taking that class on sort of a whim. After that, I took an undergrad class in signal processing. Okay, anyway, I'm going to move forward. College applications, undergraduate. Like all of you guys, I applied a bunch of places probably, right, all over the country. These dots don't apply to any particular place. They're random, so don't try to figure out what I'm talking about, okay? And so what happened to me, right? I was a good student. I took like 12 AP classes or something crazy. I had good grades. I did varsity sports, et cetera, all the things. So I started getting rejection after rejection after rejection after rejection after rejection. Sometimes they piled in a few at a time. I think I got a dozen rejections over, a month. And I was basically totally destroyed, and I didn't understand why this happened to me. But there was one place, I got one place said yes. And I feel incredibly lucky because that place actually was Georgia Tech. And so what can I say about this other than what I was interested in at the time was not engineering. I wanted to study like political science or biology. I don't know, I was like 17 years old. I didn't know what I was doing. I just wanted to go to college, right? So Georgia Tech looked at me and looked at all my stuff and they said, this guy would be a good engineer and we're going to give him a shot. And they did, and so I went there. So I'm really thankful to whatever admissions counselor looked at my stuff and said, we want this guy. So the point here that I eventually learned many years later is that rejection actually doesn't equal to failure. The way I see it now is if you're not getting rejected, your goals aren't ambitious enough. You've probably heard this before, right? But I'll say it again. But to me, there's some really important positive sides of being rejected. One of those is you can basically burn away your pride and arrogance when you're young. You think you're great when maybe you're not that great. You can avoid a lot of regrets, what ifs, if you just try, right? You never know if you don't try. And you also find your weaknesses. So I went and did a BS in Biomedical Engineering at Georgia Tech. I was like, hey, I'm in engineering school, I should do engineering. And I chose BME because of my interest in life science and in biology. As you can see from stuff I've been telling you, I love that kind of stuff. And cool thing about Georgia Tech while I was there is it underwent this incredible transformation where there's all this building, this like tremendous rise in rankings, all this hiring going on. And the same kind of thing is actually happening at UF right now. That's why I'm so excited to be at UF because it's just an energetic place that's vibrant and growing. Something I need to mention is that one of my TAs is this guy, Charlie Gershbach. He's now a professor of biomedical engineering at Duke University, very famous person now. He came up with one of the first in vivo gene editing therapies, all right, for Duchenne muscular dystrophy. But at the time I met him, he was just my TA, right? He was a grad student. And he singled me out after class and he said, hey, Ed, have you ever thought about doing research? I was like, no, right? Why would I do research? He's like, well, the research is cool. Why don't you come by my lab and I'll show you what I'm doing. And I was like, why? Okay. I mean, he's my TA. I guess he's grading my assignments. I should do what he says. Right? So I went up to his lab, and he showed me all this amazing stuff. And I was like, wow, this is cool. Maybe I should do research. He's like, yeah, why don't you send some emails to professors, see what you get. So I did that. I sent out I emailed every professor in the department. It's like, I want to do research in your lab. And how many responses I got? Like crickets, 0. Nobody responds. So I go back to him the next week in class, and I'm like, Charlie, nobody responded to me. And he's like, well, you only applied to BME, right? There's also ME and material science and all these other places. I said, oh, you're right. So I tried again, and I sent out some more messages. And what I didn't know is he secretly went behind my back to some professors he knew, and he said, hey, you're going to get an email from an undergrad student that I know. You should respond to him. He's good. And so I didn't find out about that until a long time later. So I did get to do undergraduate research with this guy, Mark Levinston, who's now at Stanford on cartilage tissue engineering, which was like a new world to me. And this was a grad student who mentored me, and I was like a mess my first time in the lab. I had no idea what I was doing, and he was very, very patient and trained me and taught me all the stuff that I know how to do now. I also did something crazy. I did a minor in a totally different area called international affairs. Anyone do a minor not in engineering out there? Yeah, right? Why'd you do it? Okay. That's awesome. Good for you. Yeah, so I've always been interested in that side of engineering too, right? And this worked out really well for me, actually. So I did this minor. I did a focus on transatlantic relations, US-EU relations. Probably not the best right now as they have been in the past. But, and that led to me getting the chance to do a study abroad in the EU. So Georgia Tech has this program where study abroad was actually in-state tuition for me. So it's cheaper for me to go abroad than to stay in Atlanta. So that was great. And that actually led me when I was in grad school to get a graduate fellowship in a program on economic policy and the societal context of technology, which was really an amazing thing that I really like to consider in my research now. And that then led me to a postdoctoral support for an international postdoc I'll talk about in a couple of slides. But the other thing I want to mention is I also met this particular individual who several years later I asked to marry me, and she's now my wife, Amanda. So you never know what's going to happen if you step outside your comfort zone. Okay, so same thing happened to me when I applied to grad school, right? Lots of rejections. One place said yes, also in Atlanta, Georgia Tech. Okay. They still believed in me. And I had the good fortune when I stayed there. I moved to the other side of town actually, that was great. I got to work for this guy, Andres Garcia, and he's been one of the most important people in my life, my Ph.D. mentor. I'm not sure why he picked me for his lab. I think I was like 4th or 5th on his list, and other people just went other places. So he's like, oh, okay, I'll take you, right? But I got to work with all these wonderful friends, great people in his lab. And Andres at the time, he was like an associate professor, pretty newly tenured. He just graduated his first crop of PhD students, was looking for a few more. He had some grant money. But now he's the executive director of the Petit Institute of Bioscience and Bioengineering and the Regents Professor at Georgia Tech. And he's also the current president of the Society for Biomaterials. So now he's one of the most important and high-powered people in biomedical engineering. And so somehow I just connected with him, and I was like, this guy has a spark. I want to work for him. So if you ever meet someone like that, try to get connected with them, because it can really help you. My PhD thesis project was to design synthetic analogs of biological extracellular matrix. So what that means in a nutshell is that your body's made out of cells, but there's all kind of other stuff in there like bone and cartilage, right? Tendon and stuff. And all that is made out of structural material, and that's the stuff in between the cells, and that's what we call the extracellular matrix. So why a synthetic one? Well, because we can control all of these things that engineers care about, like the mechanical properties, the chemistry, the delivery format, precision, right? All these little words that we love, and incorporate bioactivity. So this is what my job was as a PhD student. So I was like, okay, what we're going to do is I'm going to go in the literature, I'm going to look at like the most famous people and see what they're doing in this area. And what everyone was doing, the hot thing was, again, polyethylene glycol, and they're using a chemistry based on acrylate. And the cool thing about acrylate is you can use a photoinitiator and UV light to polymerize it. So people were like patterning it with light and all kind of really neat stuff. And so I was enamored with it. So I was like, I want to do that too. And so what we started working on was designing these materials that are these squishy hydrogels that can grow blood vessels like that. So that's what happens when you implant them. And we functionalize them with a growth factor called VEGF, which when you implant them, it causes blood vessels to grow. I mean, it's more complex than that, but that's all the time I have. So, okay, so then I was like, okay, but the major goal was islet transplantation therapy. I mentioned that before. Type 1 diabetes was the thing we wanted to cure, so we take islets from a donor out of the pancreas, and mix them with our hydrogel and put them back in the recipient to cure diabetes. So we thought enhancing islet engraftment survival and function through the use of our engineered materials. That was the goal, all right? Put it in another way, design challenge, right? You guys are in senior design, you have a design challenge. We had an engineered material, engineered matrix. We wanted to grow blood vessels into these cells we were transplanting, right? So 4 years of work, I made this material, it grows blood vessels, we put islets in it, and it was a, if I can make this thing go, complete failure, actually. It was like 4 years into my PhD, like a couple weeks before Christmas break, right around this time of year. I have my rats, I have the islets, I have everything working, and I go in there and I check on them, and the islets are all dead. All dead. Nothing worked. So I was just like, I called my boss, I was like, everything is messed up, nothing works, you know, the project isn't a failure. And so when you're in a situation like that, you want to know who your real friends are. Right? Those people that stand by you, right, when it doesn't work out, when you're down low, those are your real friends. I mean, you're going to run into people in your life who only care about you when you're succeeding. And my boss cared about me a lot that day. And he told me, you know, he picked me up and he told me, you know, everything is going to be fine. You're going to go home for the break and come back, and we're going to turn this project around. You also learn what you're made of, and you learn what's important to you and how hard you're willing to work to succeed. And the other thing that happens when you fail big time is you stop looking at others for affirmation. You start looking more at yourself. Okay, you know who this guy is, right? Anthony Bourdain. He's one of my heroes. And this quote of him I find really inspiring. So he says, if we fail, we want to fail outrageously, foolishly, gloriously, giving it everything we've got in the cause of making something new and strange and hopefully awesome. And I really took that to heart. To me, I was like, that guy is talking sense, right? That's really important. So it doesn't really matter if you fail, if you're like super proud of what you're doing in the process. And that's what I was like, I want to like rearrange my life and follow more of this principle. So what I did is I went back to the chemistry and I said, why is that killing the islets? And let's come up with something new. So what I ended up doing was changing, I mentioned this before, the maleimide groups, some of the N groups, coming up with this 4-arm type of molecule and cross-linking it into a network mesh. And then when we put the islets on it, they survived really, really well. And the reason is that the photoinitiator was generating free radicals, for those of you in chemistry, and those are really toxic to beta cells. They die. So this process is much more gentle, much more friendly, and the beta cells are able to survive. And so we could revascularize them, grow new blood vessels inside there in the green. And we put them in diabetic mice, and we cured the diabetes in the mice. So it worked. So this, by far, has been the most successful thing I've ever done as a scientist. It's got almost 300 citations now and growing. A lot of people have followed and used this chemistry after we published it for the first time. Okay, let me change gears. Let me tell you another story. This one's funny. This was my first ever research presentation, okay? So first year of graduate school, I wrote an abstract, and I submitted it to the Society for Biomaterials World Congress, which was in Amsterdam. All right, everyone in the lab was going. It was a big event, and I didn't have any data. I just wrote the abstract because my boss told me to. And my abstract somehow got the highest score in the category I submitted it to. They sent me a letter with a check for travel funds and congratulations and all this stuff. And anyway, I was like shocked. I was like, okay. So I make my slides and I go there, and when I go there, we get the agenda, which is like a phone book, right, with all the presentations, and I find my name and I'm like, okay, what room am I going to be in? Expecting a room maybe like this, because they have like all these simultaneous sessions. And guess where I am? I'm in the main auditorium. Seats like 2,000 people with a balcony and a stage and a projection of your face about 20 feet high. Okay. My first presentation speaking in public ever, I was terrified, completely terrified. And to make it worse, there was my boss sitting, and this isn't me, this is the actual place though, I found the website, right? He was sitting in the front row there, and this is the balcony there, all these other people, and it was packed, right? Because it was a special symposium, and there was somebody famous speaking right before me. And so that guy gives a wonderful talk, and then I get out there and I'm just like, oh, okay. So I'm like, okay, I can do this. You got this, right? You got this. And so I start going through my slides. I do my introduction, like boom, 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 hit all my points. I show my, my first data slide, my second data slide, and I look at the little clock they have, and it says 3 minutes have passed. And my presentation is supposed to be 20 minutes long, and I've got like 3 slides left to go. So what did I do? I don't know. I just slowed way down. And so I was just like, okay, let's look at this piece. Over here explaining everything. And everyone in the audience is like looking at each other like, what is this guy doing? Right? And to make it worse, my boss, I can see him because he's like right here, right? And he's just like, no. He's like, can't figure out. I don't know. Anyway, somehow, I think I blacked out because I don't remember what else happened. But I got to the question and answer with like 15 minutes left in my time, and so we have to fill it. So everyone is asking me questions, and I'm giving like completely idiotic answers because I don't know what I'm doing. I'm a first-year graduate student, and he's like crying. He's like, right? So anyway, I made it through that. Right? I made it through that process alive, and I'm still here. I'm still standing. Right? For those of you that like to watch that movie, Sing, my daughters love that movie. Still standing. So the thing is with public speaking is you just have to do it a lot and get used to it. I still get nervous. I'm still not the best at it. But anyway, I figure I couldn't get much worse than that. So once you've done the worst possible thing, then it can only go uphill. Okay, so again, iceberg illusion. But I want to add something to it. Iceberg illusion. This arrow, this hand, right? So everyone talks about this stuff that, what did I do? I did this, I did that. But the real secret is, especially nowadays, things are so competitive, more and more competitive than they've ever been, is you need people to help you, right? I could fill the screen with faces of people that help me. These are people that help me. And they pushed my iceberg up above the water. Otherwise, I'd probably still be underwater without their help. And I just want to point that out because I think if you're in a position to help other people, you should do it. It's really, really important. And it's really important that you yourself seek out mentors too and use them because you need it. Okay, let's move on to the next phase. Postdoctoral fellowship. I had the tremendous opportunity to go to Switzerland for my postdoctoral fellowship. Does anyone know what a postdoc is? Nobody? Crickets? Postdoc? Okay, so if you want to be a professor, how do you get to be a professor? When you make the decision, you're in grad school, I got my Ph.D., now I want to go and be a professor. Yeah, nowadays you need to go and do a training program called a postdoctoral fellowship, and that's when you're usually a full-time researcher. So you go and you establish yourself as an independent scientist. So that's what I did. And I was like, if I'm going to do this, I want to do it my way, right? And so I was like, a postdoctoral fellowship is really hard. I want to go somewhere really fun. And I knew who I wanted to work for, and that was these people, particularly this guy, Jeff Hubble. Jeff Hubble is one of the most famous bioengineers in the world. He is highly acclaimed, and he has a citation index, for those of you that know who that is, and it's like in the stratosphere. So he's a very famous scientist. And so I was terrified to email him, right? Because I was like, he's just not going to respond. He probably gets 10,000 requests a day for people to work in his lab. How am I going to do this? And so I don't know. I just sat down and I wrote my email and I sent it to him. And guess what happened? Nothing. No response. Right? Nothing happened. But I didn't quit. I said, okay. I went and found someone at Georgia Tech that had trained with him, and they didn't know me. But I said, you can talk to my boss, Andres Garcia. He'll vouch for me. Can you put me in contact with this guy, with Jeff Hubble and Melody Swartz and these other, I'll explain who all these people are in a second. And they said, sure, I'll help you. I was like, really? Why? They're like, I don't know. Somebody helped me, right, at some point, so I'll help you. So that's how it happened, actually. Someone sent an email on my behalf, and then within an hour, I got a response from him, like straight away. He was like, yeah, come out for an interview. We'll talk to you. So anyway, those connections, personal connections. If you're out there right now looking for a job, I think that's how it really happens, right? Like just responding to job applications. By the way, I should mention, I went to his website, lab website, and it said, click here for open positions. And I clicked on it, and I was like, oh, let's see what they're hiring for. It said none. No positions, right? So don't be deterred by that. I think it's really important to go out there, use your connections. If you don't have a connection, find a connection, make a connection. See if they'll help you. You never know. This is actually another scientist at EPFL, Melody Swartz. She's really big on immunology. And then this is actually another mentor that I had there, Stay named Bekoskov. She's one of the people that discovered the way that type 1 diabetes works. So I got to work with all three of these people while I was there. The research that I did there, I won't go into the details, but I'll just mention what it was. I was really curious about the immune system. So I did all this material science, kind of mechanical engineering type stuff as a PhD student. I wanted to learn how the process works on the biology side. So this is a beta cell. This is a white blood cell, a T cell. This is something called an antigen presenting cell. And while I was there, I was working out all this pathway about how the T cells become activated to recognize the beta cells and attack them. So through that experience, I gained an appreciation for the beauty and complexity of biology, right? I failed a lot at this process. I'm not going to talk about all of those failures because I don't have time, but it was really, really hard, and it took a lot of grit to get through and learn all these new skills. Life in Switzerland was great, I would say. It's like an Instagram utopia, okay? You know, this is the Matterhorn there. I was like doing all the native stuff like mountaineering and skiing, and there's a lot of cows and amazing dairy products and wine, and it's like picture perfect all the time. And I had some more milestones when I was there. My wife and I, we had 2 babies, Nora and Maisie. They're 2 years apart. There's Nora with her little Swiss cow. Yeah, so that was another thing that changed my life. Okay. So one day in the lab, I'm almost done. One day in the lab, something unexpected happened to me. I was down in the microscope room. It's dark. There's all these microscopes. There's a bunch of grad students in there like fiddling with their samples, right? And I looked in the microscope and I saw this, and I basically jumped up and I was like, yes, yes, right? And everyone's looking at me like, that guy is nuts. What is he doing? All right. And it was because I saw this picture, and to me it was super meaningful. Now, you guys, it doesn't mean anything, right? You see a bunch of dots. But this was a problem I had been working on for like 3 years, and I tried to understand something that was really strange to me. And so I put it in a nutshell for you. These dots here, this is 2 beta cells. The nucleus here is in red, so this is the same cell in each frame, just stained with different colors for different things. So this is a protein. It's called GAD65, and that's actually an enzyme. And what that enzyme does is it makes a neurotransmitter called GABA. All right, GABA is an inhibitory neurotransmitter. So anyone ever heard of Xanax before? Benzodiazepines, those work on GABA in your nervous system, on the GABA receptor. GABA is also generated at high levels in the pancreas, in the beta cells, in the islet. And what I saw was what we expect, and what all the textbooks say for a long time, is that this protein generates GABA, and GABA goes inside of these little dots. And I'll show you on the next slide what that means. And that's not at all what I saw. It didn't match it. It's completely different. So if you think about neurons, right? What neurons do, you have a synapse, neurotransmitters. You've probably seen this diagram before somewhere. Okay? So here's GAD65, little protein, making GABA, generating GABA, and then there's a transporter, puts the GABA inside. And then you have a calcium wave comes, and then it dumps the neurotransmitter outside in the synapse, and then it makes the connection to the next cell. So in the beta cells, what we saw instead was, yeah, we have the synaptic vesicles, GAD65 there, but the GABA stays outside. It goes everywhere in the cell, and but we know it's getting out. Everyone for a long time has known that GABA is released from the beta cell. But now I realized that we don't know how. And I was like, I have a discovery on my hands. Okay, and that was an amazing feeling as a scientist, the first time you actually realize you were the first person in the world to see this. Maybe it's like a microcosm, a really tiny aspect of biology, but you realize something, and in that moment, you're the only person in the world that knows that. And I was like, wait, I discovered something. And I was like, wait, what do I do? What do I do with a discovery? And so what you have to do is you have to publish it, and to publish it, you have to make a story, right? All the aspects, you have to do all the work. So like the last 6 years of my life, I've been trying to answer what this little question mark is. And I can tell you, just a few months ago, we actually figured it out. So here at UF, in my lab here. Anyway, so to wrap it up, I'd say new beginnings. University of Florida, I got a job offer here as an assistant professor. My wife and I wanted to move back. She's from Atlanta. You know, we have the 2 kids. We wanted to be close to the grandparents. So I moved from the most mountainous place in the world to the flattest place in the world. But there's beauty everywhere, as you all well know. If you haven't been to the springs before you graduate, you need to go. They're beautiful. This is Rainbow Springs. They're all over, though. I should mention, though, as an assistant professor, suddenly I have found myself in the position to reject other people, right? And that's a really precarious place to be, because all my life I've been experiencing other people rejecting me and trying to succeed and trying to get somewhere. Now it's my job to reject other people. And so who do I reject? Well, I reject other people, other scientists who write grants, and I review their grants. I'll reject other scientists who write papers, and I review their papers. Sometimes I accept them. Sometimes I reject them. I have to reject grad students that apply to our programs. I have to reject students that don't do well on exams in my class, right? And every single one of those rejections is a little heartbreak because I know how that person's going to feel. But I have to, right? Because you can't accept everyone. And you realize when you're on the other side of that, it's like, well, you have X amount of opportunities, you have X amount of people that apply, and the numbers don't match. So anyway, I do that job, but it weighs heavily on me. I think it's really important to spend time and pay attention to that. Okay, so I just want to mention one other thing. Something I've gotten involved with since becoming a professor is education outreach, and this is something that's very important to me. This is a guy in the BME department, Paul Pruitt. This is us down in Miami doing an outreach event with public schools in Miami-Dade County. And I have students in my lab all the time, K-12 students, undergrad students. So I would just say that one of the greatest joys of being a professor is having the opportunity to help other people achieve their dreams. But I learn a lot from other people. As much as I give to them, they give more back to me. In my career, I've been lucky enough to know and be mentored by all kinds of different people, different races, different creeds, different orientations, different viewpoints from my own different backgrounds. And that's been such a great experience towards enriching who I am and making me a better engineer. I just want to say I'm proud to be a Florida Gator. Thank you for inviting me for this talk. That's me and my daughter on the beach. This is a gator we made out of seaweed and seashells. And I want to leave you with a final thought. So this is Bill Watterson's last Calvin and Hobbes comic strip that he ever drew. Okay? So it's appropriate for the weather that we're having right now. Maybe it will snow in Gainesville. So Calvin says, wow, it really snowed last night. Isn't it wonderful? Everything familiar has disappeared. The world looks brand new. A new year, a fresh, clean start. Right? It's like having a big white sheet of paper to draw on, a day full of possibilities. It's a magical world, Hobbes, oh buddy. Let's go exploring. Thank you.
Just wanted to say thank you to Dr. Phelps for coming here. Can we get another round of applause for him? He's been a great sport, just going through all the different meetings with us, and I'm just kind of just being willing to give out this awesome talk today. Did everyone enjoy it?
Yeah. Oh, okay. Good. Thank you.
So we're very, very thankful for him, and we actually have a little present for you.
Thanks.
Right here.
Oh.
Boy. One moment.
Okay. What's in the bag? It's going to jump out. It's a hamster.
So,
oh, thank you.
for you.
Oh, I appreciate that. Someone getting a picture?
Yeah.
Oh, here. Hang on.
3, 2, 1.
Great. Oh, one more? Okay. Thanks. Oh,
Thank you so much.
Thank you. Thank you, guys. Thank you. I'll just add, I am on Twitter. You can tweet at me. I will respond to you. I'm trying to get to 1,000 followers because they say once you have 1,000, you're an influential scientist, right? So you guys can help me out. It's true though. Scientists have the biggest reach on Twitter of any people on Twitter because you reach so many different types of people. It's true. Yeah. Anyway, thank you.